that was loud. Okay, so muscle. Is muscle on the exam? No. Is a neuromuscular junction on the exam? Yes, because we talked about it in the last lecture. All right, you have to remember the three types, and we already mentioned this. Which one's the voluntary one? Skeletal. Which one's the one that is always found in heart? How do you question that? But yeah, cardiac. And then smooth you find around hollow organs, whether it's a blood vessel, whether it's part of the GI tract, whether it's the bladder, reproductive tract, it's around hollow organs, smooth. When you look at their characteristics, you can refer to them as striated. Which two are striated? Cardiac and skeletal. Which one is non-striated then? Smooth. Which ones are voluntary? Skeletal. Yep. Which ones are involuntary, which means you don't have control? Cardiac and smooth. When I looked at this and I was studying them originally, I thought, man, there's a lot of overlap. But what I found is that skeletal is at the one extreme all the way to the left, and I have smooth all the way at the other extreme at the right, and where's cardiac? Right in between. And you're going to find that when we talk about the physiology too. When you look at it visually, it's striated just like skeletal. It's involuntary just like smooth. Its pattern is not as regular as skeletal, so it's more like smooth. So you can keep looking back and forth. When you look at the nuclei, they're similar to the skeletal. A lot of characteristics. When we start talking about how these things contract, you're going to find skeletal is distinguished in one special way, the way that it contracts. Smooth, a very different way that it contracts. Cardiac, where would you predict it to be? Right in between. So when you're studying it, you can look at the overlapping nature and you can kind of think, well, if skeletal is all the way over here, smooth has to be the opposite of it. Cardiac is usually a mixture of the two. When you look at the level of organization, we use the terms, an organ is several types of what? Tissue, right? So when we take those tissues and break it down, a muscle organ is a lot of tissues. You've got blood vessels, you've got nerves in there, you've got muscle tissue, you have connective tissue, epithelial tissue. The cell, when we refer to the muscle cell, the other thing we're going to call a muscle cell is a muscle fiber. They're the same thing. Muscle cell and muscle fiber are the same thing. When we look at the inside of it, we're going to go back and look at those special little cytoskeletal elements. So we'll talk about the myofibrils, the fibers in the muscle. When you see myo, typically it refers to muscles. So the cytoskeletal elements we're going to fix on thick and thin filaments. And I love that they use these. The thick ones, do you think they're going to be dark or light? Dark. The thins are going to be light. So we'll refer to them as the light regions and the dark regions too. And then we're going to specifically talk about the different types, the contractile proteins. So when we talk about the actual microfilaments and intermediate filaments, we're going to use two special names, actin and myosin. So starting at organ, to the cell, to the intracellular structures, the cytoskeletal elements, the contractile proteins, we're going all the way down to the microscopic level. So you should review your anatomy. Here's your whole organ. What's a tendon made of? What type of tissue? Connective tissue. What's cool about the tendon is it's actually connective tissue that wraps around all those muscle fibers. All of this reddish pink area are the muscle fibers wrapped in this wrapping paper of tendon, and it's continuous. So the tendon forms here, goes all the way around those muscle fibers, and it stays continuous all the way on the other side. And then you have the fascia that it wraps around. Yep. So we can slice across this, and we can look at these individual muscle groups, which we call fascicles. Those are also wrapped around with the connective tissue, the fascia. And then we can take individual muscle fibers, which is another name for a muscle cell. Muscle cells aren't typically those little round things that we would learn a cell being. They're long tubes, and they can last 6 inches. So when you look at the bicep and you start taking apart individual muscle cells, there's one that starts all the way here down at the tendon, and it goes continuous as one long tube all the way to the top and the other tendon. So they're long tube-like structures. When you look inside, we're going to have all these little microfilaments. So myofibrils that are made up of things like actin and myosin. When you see the dark band, what's going to be primarily? Thin or thick filaments? Thick. When you see the light bands, it's primarily thin filaments. And these proteins actually overlap so that when you pull on them, they contract. And these are nanometers, so millionths of a meter in length. You think, well, geez, how's that move my arm up 1.5 feet? So when you pull all the way across this, it's because all of these little tiny things are working in unison. When I contract this one, I contract that, and that, and that, and that, and that, and that, all the way down. When I contract this one cell, the whole cell shortens at one time. Yep.
This is really random, but yesterday I was giving class plasma, and the guy next to me thought he was with his buddy, and he thought he was being like cool and tough, and he was like, "Watch this," and he went like that with the needle in his vein, and when he pulled his arm back down, it like destroyed his arm, and it turned into this big golf ball, but you could see part of his bicep muscle because the needle tore it.
Ooh, we have a special word for them, those people in medicine. We call them morons.
He was yelling and screaming.
I bet.
Like, am I still going to get paid? No. Never come back.
Okay, and I put this overview slide like an anatomy review. Yeah, I had a friend that donated, and when they put the plasma back in him, I don't know how they slipped, but same thing. He had a huge bolus under his arm, under his skin. Right, so when you look at this, there you have your organ, breaking it down to the cells, and then inside the cells, the actual filaments that are in there. And we'll talk about this actin and myosin as we need it. Right, when you look, the next thing you want to look at is this little structure. Remember I had dark and light bands? We're going to go down to that level. That's what we're focusing on. So with the dark and the light bands, you've got these areas that you have to know. This area is called a sarcomere. So from this little zigzag line to this zigzag line, it's called a sarcomere. It's the basic contractile unit of a muscle. So it's the basic contractile unit of this muscle. When I contract the muscle, it's happening in these nanometer space here. Teeny tiny movements. When I look at the dark section, I call that the A band. So the dark band is the A band, and you have to know that one. And we'll break this down a little bit more, and I'll say that actually the dark band is primarily something called myosin. So would you predict myosin to be a thick or a thin filament? Thick filament. The next band you want to pay attention to is called the I band. The I band overlaps two sarcomeres. So the I band starts at the end of the thick area and goes to the beginning of the next thick area. So the I band connects one end of an A band to the next A band. A distinguishing characteristic of an I band is this zigzag line right down the middle of it. The zigzag line represents the end of the sarcomere, and it's called the Z line. You have to imagine this being three-dimensional, though. A lot of people forget this is a three-dimensional. So it's not a straight line going up and down. If I looked at it, it's actually a round disc that zigs and weaves. So you have to remember this as being a tube-like structure, and this is almost like a flat plate at the very end.
So
the A band is the dark area. The I band overlaps two sarcomeres. I remember the Z line as being Z end of Z sarcomere. I know it's lame and tacky, but it reminded me that it's the very end of the sarcomere. Here's where one sarcomere starts and another one starts.
So you've got two Z lines then, correct?
You have two Z lines per sarcomere, right? And they help hold the sarcomeres together. Otherwise, when you pull, they just go and pop apart.
Oh, the I band goes from one end of the thick to the beginning.
Beginning of the next thick, yep. So it overlaps. Right down the middle is the Z. Next, you look at the zones. So you have this zone right here. It's called the hot zone is what I call it. That's how I remember it's called the H zone. This H zone, it shrinks. It's kind of a light segment right in the middle of the thick filament. And as I contract and I bring this thing closer or shrink it together, it's kind of like my fingers when they overlap. Eventually what happens, they go bink, and they touch. Right down the middle of that H zone is something called the M line. How could you possibly remember that's in the middle? I'll let you work on that. But it's in the middle of the H zone. It's also in the middle of the sarcomere. It's the center. It's another plate. This M line holds the myosin in place. So I don't know how you can relate M line and myosin, but you want to do that too. So the M line holds that myosin there. So we covered the Z line's a zigzag at the end of the sarcomere. The A band's the dark. The I band is the light one that overlaps two sarcomeres. The Z line divides the I band. Where do you find the H zone? Right in the middle of the dark band, right? It's the hot zone. What do you call that line right in the middle of everything? The M line. So when I look, the pink is myosin connected to the M line. The light blue is the actin connected to what line? The Z line. They're tied on. They're held in place. The actin is tied here. The myosin is tied here. The actin and the myosin actually overlap, again, like my fingers here, so that when they move, they don't bend, they don't scrunch, they don't wrap around, they slide. Do my fingers get any shorter as they overlap? The fingers themselves don't change length, but what happens to the distance between my palms as I slide my fingers? They get closer together. Imagine my palms as the Z lines. When you pull the actin and myosin over each other like this, the Z lines get closer together. What happens to the length of the sarcomere? It shortens. What's going to happen to the length of the whole cell? It shortens.
Where's the H?
It's a good question. So, what is this first one here? What is this space from the end of one thick to the next thick? There's the I. What do you call this dark space here? The A band. And the A band, if you look, the beginning of this myosin to the end of the myosin represents the A band. What about these two lines? What are they called? Z lines. What do you call this space in between the Z lines? The sarcomere. What do you call this line down the middle? M line. What do you call that gap in between the actins in the middle? It's the H zone, right?
Between the gaps in the
Between the actin gaps here is the H zone. So if I were to ask you a question, the set of proteins that runs down the middle of the I band and anchors the actin, what was that? So what does the M line anchor? Myosin, right? And obviously that's wrong because myosin's wrong. How about the H zone? What's stuck in the middle of the H zone? It's the M line stuck in the middle, not the actin. And how about, in fact, the H zone never has actin in it. Never, ever, ever. When the actin moves closer to the center, what do you think happens to the H zone? It shrinks. So we know that's not the H zone. How about Z line? Yep. So you got this zigzag line here holding the light blue actins. And then the A band, no. The A band is a band, it's not anchoring anything. So sarcomere, remember that's the functional or contractile unit of the cell. It's the smallest contractile unit of the cell. And then I already told you this in between the Z lines, so we're good there. So let's see if you remember. When I said the functional unit of the muscle is what? Well, automatically you wanted to say what? Sarcomere. So should you circle that and move on? Is it the area between two Z lines? Yes, it is. Is it the smallest contractile component of a muscle fiber? It is. It's all of the above. Be careful when you see an all of the above. If you knew this one and this one, but you weren't sure on this one, what do you know it has to be? It has to be all of the above. If you know for sure 2 and 3 are right, you know it has to be all of the above. Okay, the contractile components, so the individual components of this unit. You have the myosin, which is the thick filament. So where are you going to see the myosin, in the I or the A band? A band. The myosin defines the A band. From one end of the myosin to the next end of the myosin, that is the A band. Does that thing get any shorter? So does the myosin curl up and shorten up? No, the myosin stays the same length. So what do you always know about the A band? It's always the same length. The A band never changes size. Okay. When you look at this myosin, it's actually a long, twisted protein. So it's a microfilament. It's twisted and round, and actually it's an intermediate filament, sorry. It's twisted and it's round and it's curled up. I think of it as being two golf clubs that have been twisted in a vise and held together. Okay? Are they going to be really wobbly? Are golf clubs really wobbly? No, they're firm. If you took two of them and put them in a vise and twisted the two pieces of metal together, are they going to be wobbly? No, they're going to be extra firm. Is there going to be any wobble in this stuff? No, it's firm and fixed. And when I look at this as being a golf club, there's the head of the golf club. So you have this one myosin coming down here, and you have two heads poking off from the two golf clubs. The heads are important because you have two binding sites. You have an actin binding site, and I'll let you figure out what it binds to. And you have a myosin ATP ACE. What do you think this binds to? So this is on the myosin. ATP. It binds to ATP, and since it's an enzyme, what do you think it does? It breaks it apart. It breaks apart the ATP. ATP is there storing energy. This myosin head, or crossbridge is another name for it, it wants to use that ATP to bind to actin. So it breaks the ATP apart at this ATP ACE, steals that energy, and uses it to act like an electromagnet. So electromagnets are kind of cool because they're not a magnet until you send electricity through them, and then tink. This is exactly the same way. This thing's like a magnet attracted to actin, but it doesn't do anything until you give it what? ATP, energy, right. So you have two binding sites, one that binds to actin and one that binds to ATP. The ATP breaks the ATP for its energy, the actin sticks to help you contract. And we're going to go into that in way better detail. So when you take those golf clubs like I just mentioned, now you're going to take the whole bag of golf clubs and keep doing that over and over and over again. So you actually have hundreds of heads or thousands of heads on one end of your gigantic golf club and thousands of heads on the other end of the gigantic golf club. Is this thing going to be very flexible when it has hundreds of golf clubs? No. It's very firm, very fixed. But you can look, there are lots of these heads, and think of this three-dimensionally. They don't just go up and down. They go three-dimensionally all the way around this structure. So they encircle this round structure. So what do you know about myosin so far? So you know number one's true. How about number two?
No.
Nope. Because the I band is what? Light or dark?
That's light.
That's the light band. You know myosin is a thick, dark filament. You know that one's wrong. Number two or three is the primary protein in the thin filament.
No, that's actin.
No, we know it's thick and right. Actin is the thin. So we can cross that one out. Number four, change length during muscle contraction.
No.
No, it doesn't change length. Is it flexible? Does it bend? Does it wrap? No, it stays fixed. What do you know about the A band? Since myosin stays fixed, what happens to the A band? It also stays fixed. So number one had to be the right answer. Okay, next. So when you look at this three-dimensional structure, don't forget it's three-dimensional. Here you have a large, thick filament. What must that be? It's a myosin. Here's a little tiny thin filament, so they must be actin. So here's that myosin, remember, with all those golf club heads poking all the way in three dimensions around it, so that it can grab any of the actins that surround it. It's constantly grabbing actins. These golf club heads, they can grab an actin here, grab one here, and they just keep pulling back and forth. When you look at the proportion, which is there more of, actins or myosins? Actins. There are more actins than myosin. So the actin surrounds the myosins. Okay, actin, the thin filament. Actin actually crosses over into the A band, but you see most of it in the I band. It crosses over into the A band because it has to sit on top of some of that myosin. So the myosin can grab it and pull it in. You have two regulatory proteins, tropomyosin and troponin, and then you have some special subcategories. So the first one, tropomyosin, not the same as myosin. It's just called tropomyosin. And the other one's called troponin. Tropomyosin is kind of like a protective covering. Troponin is kind of like a hinge. I'm glad I wore a tie today. This is always better for my example. So on troponin, you have three binding sites. You have a binding site that grabs a hold of the tropomyosin, a binding site that grabs a hold of the actin itself to hold it onto the actin, and you have a third one that binds the chemical calcium. And I'll show you what I mean. So what did I say troponin was like? It's like a hinge. Here's your actual actin. Actin looks like a bunch of pearls, and then you form this pearl necklace, and you twist the pearl necklace. Is this going to be really strong? If you take a pearl necklace and tug on it, what happens? It breaks. Actin's fragile. That's why you need so much actin compared to myosin. It's very fragile. So it's like playing tug of war, but instead of having one thick rope, you have a bunch of little small strings that you're pulling on. Right? So actin molecules, these protein globules, they start forming this long necklace-like structure. You have these little tiny dark green spots. These are called the myosin binding spots or myosin attachment spots. What do they want to attach to? Myosin. Remember, actin, you had an actin binding spot on the myosin, now you have a myosin binding spot on the actin. They want to come together and they want to stick like magnets. So if I left those exposed all the time, what do you think they would want to do? Stick to myosin. Yeah, I'd always be contracting my muscle. So I need to protect those spots. That tropomyosin is that protective covering. It's like taking a pearl necklace and then taking a little string and covering those active spots on it. So if I have something in between these two, myosin and actin, I can't stick them together. What do you think is going to happen to the muscle when that's in place? Is it going to contract or relax? It has to relax. The actin and the myosin can't bind, so they can't contract. That's the tropomyosin. This is the tricky part. The troponin's like a what? It's like a hinge. When I put the troponin on here, the troponin holds on to the tropomyosin. It has a calcium binding spot, and another part holds on to the actin. It's like my body is the actin. My arm would be the hinge, which is troponin, and the end that holds on to tropomyosin is holding my tie. The buttons are my myosin binding spots on the actin, right? This is my actin. Here's where the myosin wants to bind. Can the myosin bind right now? No, when I have my tie covering the spots, the myosin has no idea that the spots are there. So what has to happen is I have to hit this hinge, and what's a hinge? The purpose of a hinge?
Open.
To open something, to move something. So the hinge pulls open. Can the myosin bind now?
Yeah.
Sure can. So it's like that. Take a guess at what the chemical is that hits the elbow hinge and opens it up. Calcium. Remember, back here, this part of troponin is bound to actin. This part's the calcium binding spot, and this part is held on to tropomyosin. 3 structures on the troponin that were all listed here for you.
So it's a hinge that allows myosin to bind?
Yep, it's a hinge that moves tropomyosin. When the hinge moves the tropomyosin, then myosin can bind to the actin. And this is the process that shows in your book. So right now, can myosin and actin bind? Nope. That hinge has to move the tropomyosin out of the way. What's the name of the chemical that binds to troponin to move it? Calcium. So here you see, from a side view, calcium binds, it pivots the tropomyosin out of place, and now myosin and actin can bind together. So now they can stick.
So tropomyosin, you said when actin and myosin can't bind, so the muscles relax.
If tropomyosin is in place, you can't contract. Calcium has to be present and move that out of place. And this is you can watch the process. So here's the little golf club head. There's the ATP that binds to it. It's charged now. Calcium comes along and plugs into that troponin. What's it going to move? Tropomyosin. Moves the tropomyosin out of place. Now look what happens. That electromagnetic head is going to grab a hold and pull. So you get this pivoting action. This one pulls back, and then this one will pull back. It's like tug of war. One grabs and pulls while the other one gets ready and grabs and pulls, and they just keep doing this. Pulling that sarcomere closer and closer and closer together. And this happens in millions of tiny little heads inside a muscle. Every time you just want to contract. You want to move your lips, you want to move your eyes, you have to go through this process repeatedly to contract that muscle. It's like a tiny tug of war going on in there. Okay, so we'll end on this. The cross bridge of myosin binds to what 2 things? The cross bridge of myosin binds to what 2 things? So does it bind to calcium? Calcium binds to what chemical? Troponin. We know that one's wrong. How about ATP and actin? Yes. What do we know is wrong about this one right away? Calcium. So you know 1 and 3 is wrong. How about troponin and tropomyosin? What binds to those 2 things? Those 2 things bind to actin, right? Not myosin. There it is, 229. We're out of here. Remember, Tuesday when you walk in, you'll have an exam. 65 questions, multiple choice. Right after you get done taking your exam, you have 1 hour. What are you doing? Taking the group 1 for extra credit. Don't forget, lecture quiz number 3 is due by Tuesday. If you turn in homework on Tuesday, I have it graded if you want to take a peek at it. Otherwise, yeah, drop your homework off on the way out the door.